Hey guys, welcome back to my channel. It's B for Bio. So in the previous video we have discussed about the limb field, and in this video we will be discussing about specifying limb field, and which is achieved by Hawksian expression along the anterior to posterior axis. So limbs of different vertebrates differ with respect to the somite label at which they arise, but their position is constant with respect to the label of Hawksian expression along the anterior to posterior axis. So in different vertebrates, the limbs placement might differ with respect to the somite number, but it's constant with respect to the Hawksian expression label. Hawksians in tetrapods, there are 4 clusters of Hawksians grouped A to D, and each group has 1 to 13 number of genes. And their differential expression and timing in the lateral plate mesoderm determines limbs placement. For example, in these 3 species of birds, one is the zebra finch, chicken, and ostrich, their differential expression of Hox B group of genes has resulted in the variation in their positioning of the forelimbs. So the different durations of expression of Hawksians in the lateral plate mesoderm determine the forelimb placement. For example, the anterior Hox B genes like B1 and B4, and the posterior Hox genes which are B7, B9 and B13. So the anterior Hox genes expression differ in the 3 species of birds. For example, here in zebra finch forelimb, the duration of the anterior Hox genes, that is the red yellow region, is quite shorter in comparison to the chicken, which is the longest in ostrich. So this places the forelimb in different positions. For example, in zebra finch it is in the 10th vertebrae, and in chicken it is the 15th vertebra, and in ostrich it is the 18th vertebra. Coming to a transplantation experiment of different regions of the presomatic mesoderm to the limb field, which causes change in the limb size. So there are 2 experiments. In experiment A, there is transplantation of somites from limb to flank label. So the donor's somites in the limb field are transplanted to the recipient non-limb or flank region, thus expanding the limb field. In second experiment, where the transplantation of somites from flank to limb label, that is the reverse experiment of the previous one, here the somites from the non-limb area or the flank region are transplanted to the recipient's limb field, thus decreasing the region of the limb field, and the results obtained were compared to the control site, and we can see that in experiment A, the limb bud is larger than the control site, and whereas in experiment B, the limb bud is smaller than the control site. So that by expanding the limb field, the limb bud is also becomes larger, and by diminishing the limb field, the limb bud also gets smaller. So from here, we get to know that the Hawksian expression domain causes the paraxial mesoderm in the limb forming region to be different from all other paraxial mesoderms. So the somites where Hox gene is expressed is different from the other somites where it is not expressed. And the paraxial mesoderm from the limb forming regions or from the limb field promotes limb bud formation, whereas the paraxial mesoderm from limbless flank actively represses the limb formation, which we clearly saw here. The non-limb somites repressed the size of the limb bud in the transplantation B. Now coming to the attribute of the limb field, that it represents a harmonious equipotent system wherein a cell can be instructed to form any part of the limb. So that if the limb field is split into two parts or used a barrier, and any half can form the entire limb bud. So first is any half of the limb disc is able to generate an entire limb when grafted to a new site, and another attribute is splitting the limb disc virtually into two or more segments and placing thin barriers in each segment develops into a full limb. So we can see an example that occurs in nature, that is in case of multi-limbed Pacific tree frog. Here is the skeleton of the frog, which has multiple hind limbs, and this is the result of infestation of the tadpole stage developing limb buds by a trematode cyst. And this parasitic cyst actually split the developing limb bud in several places. So this pieces of the limb bud, every piece gives rise to an extra limb, thus giving rise to a multi-limbed frog. So that's it for today. Thanks for watching. Give it a thumbs up if you like it. Do subscribe and hit the bell icon.